Welcome to Something About Science. I'm one of your hosts, Danielle, and today we have a conversation on a topic that intersects health, genetics, and future generations. We'll be discussing the fascinating research conducted by researchers at the Karolinska Institute about how Polycystic Ovary Syndrome, or PCOS, can affect not only women, but also future generations of men. In this interview, we had the privilege of speaking to two distinguished researchers about their research that challenges the way we think about polycystic ovary syndrome and its wider implications. So, without further ado, let's delve into this fascinating conversation.
I have been studied PCOS for, I think it is soon 30 years. And from the beginning, it was very much trying to ease their symptoms. But then more and more, my focus have focused on trying to understand the mechanism behind the syndrome. So my research has always been translational. So I have done both clinical studies and using different animal models and develop different animal models depending on the purpose of what we want to investigate. And along this, more and more, we use this model that is in this paper that we call prenatal androgen exposure, which means that we mimic the pregnant woman with PCOS. We know that PCOS has a strong heredity. As we shown in our previous paper, that it is 5% at least develop PCOS at adult age. But I would say that I think that number should be the double, because what we use there is actually our Swedish registers. And we are not diagnosing, and they are not getting the diagnosis. So with that said, then there are not so many models investigating the pregnant PCOS woman. It is this in mice, the prenatal androgenized model, but quite a lot of work have been done in the rhesus monkeys and in sheep. We do have rhesus monkeys at KI, but I have never worked with that. I don't think you have been involved in any of this research either, Qiaolin. No.
Oh, not monkey.
No. But anyway, so before this study, we did quite a number of studying both rats and mice that we exposed the pregnant dam for androgens during late pregnancy to mimic the pregnant PCOS woman that always have higher androgens during pregnancy compared to women without the syndrome. And before this, we have demonstrated that the offspring, they develop a PCOS-like phenotype. They become more obese, they have irregular cycles, and they also get some pregnancy problems and metabolic features, insulin resistance and adipose tissue dysfunction. But none had really studied whether it could be transmitted to second and later third generation that is not exposed. So I think the beauty of what we have done here is that we can really try to disentangle what happened in the humans, that we always have genes blurring up. Here we can really study what happens with this exposure to the germ cells, to the somatic cells, and how it is transmitted. That is so. We are continue with this. It's always like this. When you do one study, you more or less you have already placed that behind you, and now you continue with other studies. But it is there is much more to do.
Yeah, brilliant. Would you like to?
Yeah. So for me, I mean, I'm much younger than Lisa, so that's not like 30 years to research. Yeah, so my background.
She's the next generation. She's the second generation.
I get mentored. That was nice. And so my background is a development biologist and a stem cell researcher. So when I started my lab 2015, so almost 10 years ago, so it is the interest is the germ cells. So because I really interested to see how our genetic epigenetic kind of heritage can be transmitted to the future generation because I'm also development biology. I really interested in the embryo development, like cell type specifications in this case. So when I started the lab, so one thing is besides the germ cell, there's more and more this kind of development origin of health and the disease. So a lot of attention also lay on this epigenetic inheritance, which not only like a genetic material and can define, or affect the future generation, is also we are exposed to this uterine environment, which because during the development is really plastic or a development plasticity, we call it, because we really respond to a lot of signals to change the gene expression. So that is major, like two very strong interest when I started my lab. So. And I think me and Lisa still thinking, try to remember how we hook it up actually. So, but
I think actually it was then because Qiaolin was at another department. So it was then the head of the department came and talked to me. Yeah, we have this young researcher that is interested to join the Department of Physiology and Pharmacology. Yeah. Would you be interested in this? And then I checked her CV and what she had done. I was like, yeah, wow, yes, of course. Take her here.
Because by that time, we are using this single cell sequencing technique. During my postdoc, I was working with Richard Sandberg, who is like the inventor with the SmartSeq, basically technology for the single cell sequencing. So I've been using it for analyzing the early like blastocyst kind of cell lineage specification. So I was really interested in the maternal uterine environment, and Lisa has done like quite a number of like research, especially showing like maternal like hyperandrogen environment really affects the next generation. So I think we already started this like 3 generations. So I think it's really nice to really follow this, to see what is like behind this epigenetic inheritance. As said, so we use pure genetic mouse models, so then we can really tease away this genetic influence, really look at how the epigenetic affects. So we started collaboration since then. I think since I moved to the FIFA department, like 2017 something. And so I think this line has been always our strong, both like interest from both sides. So we have been kept. That means we start from the female progeny, and we published that. I think that was also, we published much in impactful journal like Nature Medicine, but we are also thinking that's not like received that much attention until now when we published in the male progeny.
But honestly, it did reach a lot of attention.
Yeah, it is.
But I think also it is
more like a broad impact, because maybe if you PCOS mother, then their daughter also have a PCOS, maybe something people think is not that strange. But now you also affect the sons and their sons, then I think something really probably out of people's expectations. So that's why I think probably this is more impact. And
because it is not the first time I have published on the male offspring. I have had 2 PhD students that actually have defended their thesis about it. So honestly, I hope it is not because of this male offspring we are studying here, because I can see also from the general community, the femtech is rising. So it is an awareness of female or women's health that is tremendous. True. I don't know what happens here around because it's like everywhere.
Recently, like both science and nature actually really highlight this both reproduction and the woman's health. So highlight it is underfunded, and actually their impact is actually much huge. I mean, we know like their offspring also, like offspring's offspring. So it is nice. Maybe also as you said. So now it's also timing, kind of the awareness and everything. Just yeah. Mm-hmm.
So. Something I would like to highlight that what we have done, which I think is something we continuously in our clinical studies, we always do ask them to fill in questionnaires, asking for their health-related quality of life, anxiety and depression, and they are more depressed. And it is not really driven solely by their excessive weight, because PCOS is closely linked to obesity, and maybe obesity can even cause PCOS, although not the other way around. So PCOS does not per se, from a genetic point of view, cause obesity. So we did first study that we used this prenatal unionized model in rats and co-treated with flutamide and tamoxifen. That is, we gave testosterone in that study, so that can be aromatized to estradiol. So we gave both testosterone and flutamide and testosterone and tamoxifen, and we could see that both in female and in male offspring, they develop an anxiety-like behavior, which can be reversed by both flutamide and tamoxifen. And we could really see that we did ICV, when you do intracranial injections of testosterone. So we didn't have it on whole body level as we have here. And by injecting testosterone for 8 days, they develop an anxiety-like behavior. We did that only in the female offspring. So that is also something that there is a big need to further discuss this. Also, their body dysmorphia.
Body dysmorphia.
Yeah, thank you. And so on. So yeah, there is so many things to do, and we try to address each aspect.
Yeah, brilliant. Honestly, I think we've got everything we need there. That was so thorough and helpful. So thank you so much. We'll definitely be able to get some really good snippets from that. Have you got anything else you wanted to add, or do you feel like you've covered everything?
Yeah, I think for me and also for us, we are really interested in the mechanism, also more like really why this is happening, and how this uterine environment can change this development trajectory of the embryo development. I think both of us are very interested to maybe more intervention with understanding the mechanisms, and then we are starting we're also collaborating, studying the placenta's role I think in the future kind of research. We are trying to do more like because we are already published. Also Lisa, published a lot of this. I think evidence-based research. So I think we are more and more strongly believe it is kind of inherited in more than one generation. So now can we do something about it? So I think for our future research is really to test part of the uterine environment where the placenta's programming all the really germ cells. So if we can before the conceiving the both oocyte and all the sperm are exposed, especially if it's maternal. Yeah, the oocyte has been exposed in this follicle uterine environment. Is the oocyte has already been kind of affected. Can we do something about that? So yeah, I think that is the more also our research kind of direction in future to really help this and do something about it.
In the clinical studies that I have ongoing now, we actually collect tissue samples and whole blood and serum and we actually collect free tissues muscle, fat, and endometrium from the same women, that we can do really detailed molecular analysis on single cell level, which I think will really change the field, because it has the possibility to really detect true drug targets, so we can target the treatment, and also potential biomarkers. So I think that is something, I think it's very important to find markers so we can actually detect it early and advise how to live a good life as a PCOS girl and woman later on.